So for the methodologies, I just wanted to have an overview for the high school student. So we collect the samples in the natural habitat. For example, we collect the different substrates. We do it, incubate it. We observe it using the macro and micromorphology of the fungi. And we get the characters of the fungi that is very important in the description, especially when you want to introduce a new species, new genus, new family, or new order. We did single spore isolation also. So we will make sure that what we are isolating is aquatic fungus, not from terrestrial environment. We incubate the culture to grow, but aquatic fungi are very slow growing. So we need to be more careful because they are easily to be contaminated if we are not careful enough. So we did photoplate preparation for the description. This is very important, especially our center in Thailand. We are more of like morphological and phylogenetic analysis, and we are very little strict when it comes to the morphology and the photographic plane. We did the colonial description, and for the colonial description, we also get the morphology and culture. So this is very important when you want to have a sexual and asexual morph connection of aquatic fungi. And after that, we do molecular analysis to find the phylogenetic placement. And this is also very important also if you want to introduce novel taxa or new species to science. So these are the substrates that we usually observe. So you can see here, I'm so sorry. Here we have in the marine environment, we collect wood. For example, there are also from the mangrove habitats, seeds, pneumatophores, also from the prop roots, leaves, submerged, etc., etc. It should be submerged because if it's not submerged for a quite long time, we cannot isolate aquatic fungi. And for us, because we already know the morphology of aquatic fungi, we can distinguish, oh, this one is an aquatic fungus because of this belongs to genera. We also isolate from salt marsh environment in Thailand and also from UK. Sediments also. You can see here, this is a fruiting body of fungi. And also from freshwater environment, we collect woods and grasses. I'm so sorry. Parang hindi nag-next. Okay. After that one, when we collect the samples, we do morphology. So very important here, we use compound and dissecting microscope to obtain the micromorphology and the macromorphology of the fungi in their natural habitat. We don't do plating. Usually when I did my master's, we do plating, but here we need to observe them in the substrate because they have different morphology and usually they have the different morph stages. And after that one, I can draw. So for example, we observe, this is a fruiting body. We checked for the fruiting body, we cut it. So when we cut it, we can determine if it's a sexual morph. Just take note, this is sexual morph or the teleomorph. This is what we call, this is ascomycete. Sorry, cannot see. No? For the asexual morph or the anamorph, we have the hyphomycete and this is the coelomycete. So what's the difference? Ascomycete, these are the following structure. They have the asci, ascospore, and the whole ascomata. For the hyphomycete, we have the conidiophore and the conidia. For the coelomycete, we have conidiogenous cell and the conidia and the conidiomata, the whole fruiting body. I will not further discuss this one because it's a long discussion, but I just wanted you to see the different morphology of aquatic fungi. And after that one, we cut with the razor blade manually. So vertically or the cross section of the fruiting body. And for hyphomycete, we just pick it up. For the ascomycete, the asexual morph, we cut it and put it in the slides. So we put water, put the fruiting body there. Okay, so you should be careful we don't have bubble, because if you have a bubble, it's difficult to see the morphology and you cannot see the whole character of the aquatic fungi. Okay, well, sorry, it's not going to the next part. What is this? Okay, after that one, when we do the cutting of the observation, we observe in the microscope, and these are the morphology. So it's quite interesting, right? So here is the hyphomycete. You can see the conidiphore. It's like a small forest on a very small wood twig, and you can see the morphology of the conidia. This is the conidia. See the spore. Long time ago, we just see the spore as like globus or circular, things like that. But in the natural habitat, they have different morphology. This is a coelomycete. Coelomycete, you can have the whole fruiting body here. This is the conidiomata, the wall, and you can see the conidiocinous cell. This is very important when you see when you wanted to have a detailed morphological description. And this one, we synonymize, we introduce this as a new combination. This one we introduce as a new species to, yeah, this is Halobisothysium from the genus Halobisothysium, this one from Sodobactrodesmium. So here for the dissecting microscope, you see the morphology in the wood. For the compound microscope, you see the micromorphology of the aquatic fungi. For the sorry, how can I? Okay, for this one is ascomycete. This is the sexual morph. This is very important also when you want to observe the different stages. For example, the immature to mature stages of the asci. These are the spores of the fungi. Here, different species also. These are new species that we introduced from Halobisothysium and from Sarcophora. So here again, the morphology in the dissecting microscope is here. And for the compound microscope, you can see the very beautiful morphology of the ascospore and other morphological characteristics. When you observe already, you need to obtain culture. One way to do that is to do single spore isolation. From the word itself, you need to only obtain one spore that is germinated and transfer this to another new petri dish. It's very important when you want to have a good molecular data. So what you're gonna do, just mark the petri dish, cut the section, the fruiting body, pick it up with a sterilized needle, pick these are the spores, put some water. This is sterilized water in the sterilized, what is this? I forgot the name, with a cover slip, glass slide, sorry. And this one, you need to put all the fruiting body here, the one that you cut, crush it to release the spore. So pipette it out. You need to pipette it out and put in your graded petri dish or media plates. So incubate in room temperature for 1 day, but for aquatic fungi, usually it takes 2 to 3 days for them to germinate because they're very slow growing. And sleep. I will sleep then. After the next day, we need to observe. And for example, we have germinated spores. You can see here, you need to observe in the compound microscope. If you found it, go to, transfer it to another fresh medium. If you have no spore germination, just cry, joke, and you don't have data, just cry. But the thing is, you need to use another different culture media and repeat the process again. That's quite tricky and very depressing also when you work on aquatic fungi. But it's okay. It's an interesting field. Sorry, these are already germinated spores. So you can see here the different germ tube. Why are we taking pictures of germinated spores? This is very important when you want to see you want to see you want to see the pattern of where the germ tube arises. For example, this one, it arises from the basal part. Some of them will arise on the different angle, and some at the polar and at the apical and basal cell. So this one, you need to pick it up, cut again. These are the spores, and you need to be more careful because sometimes some aquatic fungi have a lot of fruiting body also in the substrate. So you need to familiarize yourself of what you are observing. So we need a muriform spore, right? This is not the cylindrical one, so you need to pick this one up. And you need to be careful also because sometimes maybe if you're not good enough or maybe you're tired and you will pick this up because they're very small. They are, you can see only using microscope. So you need to be more careful. And after that one, take the picture and transfer to another plate. When you transfer to another plate, this will be the picture. Quite interesting. So for me, because they're slow growing, I put 4 or 6 in one colony. And you can see here, this one are aquatic fungi with the same morphology. And this one also, this is another like a pure culture already of aquatic fungus. You can see also the sporulating structure here. After that one, we do the photographic plate description. So we need to be concise with the morphology we wanted to share. For example, if you want to publish, this one is a new species. So if you want to publish it in a good journal, you need to have a good morphological description also. And one thing we need to do also is to observe the culture. And this is very important when you want to have a asexual morph connection of aquatic fungi. So these are the cultural observation. When you say cultural observation, we observe them from the culture. These are the chlamydospore. This is a new species, Cladophila aquatica, we introduced. And this is a new host record, Sauriella nipae. You can see in the culture here, these are the cultural micro characters. It's quite the same, but you can see the different stages from mature to immaturity. See, they become hyaline, then they become like dark brown at the end, pigmented already. And after that one, when you have a good enough mycelia already or culture, you can do a phylogenetic analysis. This is just a step, but this is very easy also. So you have DNA sequences, you need to blast to see the closest relative. You need to assemble the forward and reverse data. You need to assemble your multi-gene data set. You edit it, you do alignment, you cut it using Chimal, you do conversion for further analysis. This is for the maximum likelihood, Bayesian, and the maximum taxonomy. And after, if you have this one, you can have a phylogenetic tree already. And you will visualize, revise. You do not revise it like revise the whole tree, but you only revise the name. Sometimes when you do analysis, you have the code, right? So the correct name of the fungi. And you do further analysis to determine if it's new or not.